Hello student, today I'm going to start a new topic, a new cell organelle called mitochondria. As you know that mitochondria is said to be the powerhouse of the cell. Powerhouse of the cell. Why they are said to be the powerhouse of the cell? Because they produced energy. Okay, a cell required energy for its activities. So this cell organelle, they produce energy in the form of ATP. Okay, in the form of ATP, and this is an energy. I'll write E for energy. Okay, there are two different processes that involve, that is ETS, electron transfer system, and oxidative phosphorylation. Oxidative phosphorylation. So this ATP were produced by these two processes during the process of respirations. And this is the reason that they are considered as the powerhouse of the cell. Now this mitochondria, okay, was first observed by Colliker. First observed by Colliker in flight muscle insect. Later it was named as, phyla by Fleming. And there was another scientist who termed the name as bioplast by Altman. Okay. Later, the term mitochondria was given by Benda. So it was first observed by Colliker. In flight muscle cells. Okay, phyla by Fleming. The term phyla was given by Fleming. The term bioplast was given by Altman, and mitochondria term was given by Benda. Now, the numbers of mitochondria have been observed in different organisms. Okay, for example, one mitochondria for one cells. Okay, mitochondria per cells. Per cells, it will be one mitochondria. Cholera, Chlamydomonas, okay, example like cholera and Chlamydomonas. Now in the flight muscle cells, okay, flight muscles cells, around 5 lakhs of mitochondria was observed. See, and 50,000, okay, in giant amoeba was observed. Now this variation in the number of mitochondria, okay, depends on the activity of the cell. Okay, if a cell is more active, the numbers of mitochondria, there will be more numbers of mitochondria. Since the muscle cells are more active, you can see that there is a very large number of mitochondria. Now, when mitochondria was observed in an electron microscope, okay, not the compound microscope, but an electron microscope, they found that the mitochondria has a double membrane, okay, and they also says that it is a semi-autonomous organelle. Semi-autonomous. Now, what does it mean? Semi-autonomous organelle means this, your mitochondria is said to be semi-autonomous organelle. Why? Because they have a DNA, its own DNA. Now you know that DNA can multiply, they can replicate. If it has its own DNA, it has a circular DNA which they can replicate. Replicate means they can multiply. Okay? But they dependent on nuclear DNA. Since nucleus also have its own DNA, for its processes, for some protein, okay, for some protein, the mitochondrial DNA, since mitochondria has its own DNA, they will replicate, but for some protein, they depend on nuclear DNA. And second important thing is that they also have protein, ribosomes. So this is the reason that they are said to be semi-autonomous organelle. Now, as I said that when observed under electron microscope, it has a double membrane. So let's discuss the structure. This is a very common structure that you will find in your textbook. Okay. So I made a membrane, since it is said to be a double membrane. So I will consider this as outer membrane. Outer membrane. This outer membrane, okay, this outer membrane have less protein. Protein is very less, but they are permeable. Its membrane is permeable, okay. Now there is some fold, okay, like this. There is some fold. There is some fold, and that fold is said to be considered as inner membrane. Another inner membrane. So it is said to be double membranous structure. Okay. Now this inner membrane have more protein. Okay, and more protein, and but selectively permeable. Very selective. They allow the molecules or ions that is required in a selective manner. Okay. Now, this fold-like structure, you can see this fold-like structure. This fold-like structure are said to be crystal. Okay. All these folds-like structures are considered as crystal. The space between this outer and the inner membrane, you can see that there is a space between the outer and the inner membrane. Okay, this space is said to be outer compartment. Okay, this is said to be your outer compartment. Now, inside you can see that there is a space, so this is said to be your inner compartment. Okay, I'm repeating once again. The mitochondria has a double membranous structure. The outer membrane, less protein, permeable. They will allow the molecules or ions to pass. Inner membrane, they have more protein but selectively permeable. The space between the outer and the inner membrane, the outer and the inner membrane we call outer compartment, and inside, okay, inside the inner membrane we call as inner compartment. Now, inside this inner compartment, there is a fluid called as matrix. Okay. Now this matrix they are usually pale yellow in color. Pale yellow in color. Because of a pigment, they have a presence of pigment called as riboflavin. And because of this, they are pale yellow in color. Now this matrix also contain enzymes. They have useful enzymes, okay, for the processes of required for the process of cellular respiration. And this is one of the functions of mitochondria. Okay, they are the site of respiration. Mitochondria provide the site for respiration. So since they contain enzymes, now they also contain divalent ions. Divalent ions in the form of like manganese, okay, iron. This is divalent ions. Now this matrix, another point just right here, this matrix have a circular DNA. Circular DNA, and they also have some ribosomes. Circular DNA and ribosomes. Now this circular DNA is, okay, I'll make it here, circular DNA. You know that the DNA is composed of nitrogenous base, right? Like adenine, thymine, guanine, cytosine. But in circular DNA, the mitochondria which has a circular DNA will have GC rich content. Means they will have more number of guanine and cytosine, okay? And because of this reason, since they have triple bond, guanine and cytosine, you can see there is a triple bond, okay? Because of that, they have high denaturation step. Denaturation, I would say as step or temperature. To denature that strength, we need high temperature to break this bond because of the presence of triple bond, okay? Whereas ribosome is said to be 70S. See, 70S, you can take an example of the prokaryotes, right? So that's why scientists, he reported that, he come up to an idea that maybe that since this mitochondrial, they have 70S ribosome, okay? Because of that, their origin may be prokaryote, okay? Or maybe symbiotic, that was proposed by Altman. This, as you know, that these ribosomes are responsible for the production of protein synthesis. So mitochondria has its own mitochondrial, has its own circular DNA, okay, and ribosome. Now, this is about the matrix. Next important component is the crista has another specialized structure, knot-like structures, and this is considered as elementary particles. Elementary particles, or F0-F1 particles, or oxysomes. Okay, you can consider as elementary particles F0 or F1 or oxysomes. Now these particles, okay, has two. One is head and one is tail. H0 is said to be head and H1 is said to be tail. And this tail part have ATP synthetase, just right here. The tail part, that is F1, has ATP synthetase enzyme, and this enzyme is responsible to help, okay, this enzyme is responsible for the synthesis of ATP. Okay, so now this is a brief explanation about the structure of mitochondria. Now the functions of mitochondria. Okay, in one or 2 points I would like to share. That is, the first point is they are considered as they produce ATP that a cell requires for its various activity. The second is that they also help. Okay, they are the site of the cellular respiration. Okay, these two function is very important. They are said to be semi-autonomous organelle. Why? Because they have a DNA that can replicate itself, and they have a ribosome which is useful, which can synthesize its own protein. Okay. Now in the next class, we will discuss about another cell organelle known as endoplasmic reticulum. Thank you.